Hello. So I got into cancer or medicine because of my family. My mother died of lung cancer in her 50s. My only aunt, early 40s. My only cousin, breast cancer, late 30s. Oops. How do I get to advance? Oh, there we go. So, the test we have is much more than a concept. We have over 20 clinical studies, 7 countries, over 10,000 patients. Most importantly, you'll see in this talk, it's not that we find cancer early, we can even detect precancerous tissue. Over 40% of the cancers, lung being the biggest, and 20% are non-smokers. Actually, the inventor of the test, who's one of our founders of our company, his dad was a non-smoker and died of lung cancer. In fact, 1 in 4 of us will be diagnosed with one of these cancers. 1 in 4. Approximately 80 million Americans. How do our tests work? They're actually very, very simple. It's a complex technology, but we got it into a single strip. Point of care, you'll know within about 15 minutes. Basically, for the lung, all you have to do is cough up some sputum, put it on the stick, and it's like a pregnancy test. In about 15 minutes, there's color or no color. Breast, so you have to take a sample from each of the areas, like a breast pump from the nipple aspirate, colon rectal swab, prostate, prostatic fluid, and uterine cervical mucus, just like a second pap smear. And again, it takes about 15 minutes. If you get this slide, you get the whole concept. You understand how enormous and how significant this is. You can see in the piece of colon tissue the cancer, and you can see to the right what looks like normal tissue. Our marker is present away from the cancer as well. So you can see in this case, with the cancer, it tests positive, and it tests positive away from the tumor. Healthy patients, they test negative. So our marker is a altered sugar in the intestine, or lung, prostate, etc. Here's a colon cancer trial. All the patients tested positive, 100% sensitivity. It's outstanding. Almost 96% sensitivity or specificity. But there's a much bigger implication to this study. After removing the tumors, going to the next one. I need to go backwards. 60% of the patients tested positive after the tumor was removed. Tested positive. Within a year, there was a number of recurrences. So what it's doing is it's finding the marker when the person's actually cancer-free. The people that tested negative after having their cancer removed, no recurrences. We've had a similar trial with lung cancer. The opportunity, it's huge, of course, right? 1 in 4 of us, a couple of the competitors, liquid biopsies, they don't have the same sensitivity, it's lower sensitivity, which makes sense because when there's a small tumor, there's going to be very little DNA in the blood. But one of them, Thrive, sold for a couple billion in 2020. Grail sold for $8 billion last year. So I love the liquid biopsies because they can screen for many cancers. But when it comes to the main cancers, the deadliest cancers, I think we need our test. In fact, I know we need our test. Market potential, for every 1%, it's over $300 million. And these are very conservative prices. We put a price of $195 for 4 tests for women, lung, breast, colon, and uterus. 3 for men, lung, colon, and prostate. My guess is the price of tests will be a bit higher, but we want to keep them so everybody can afford them. But that's over $300 million for just 1%. The competition with lung, low-dose CTs, they still emit radiation of 14 X-rays. Mammogram, 4 X-rays. Prostate, the PSA does not differentiate from enlarged prostate from a cancer, whereas our test does. But in the prostate, we've only done lab, but in the lab, it completely differentiates perfectly between a cancer and an enlarged prostate. Uterine, it's a terrible void in medicine. There is no test for uterine cancer. Colon, I like the Cologuard test, but it's expensive, and a lot of people don't want to. It takes time to get your results back, a couple of weeks, a week, a couple of weeks. Who wants to collect their own stool? And the liquid biopsies, again, the limitation of them is the sensitivity for early stage cancers. When it comes to imaging tests, you can see, I showed you that on the intestine, a normal looking intestine, you're not going to be able to see it if precancerous. So imaging tests won't find it early stage. Same with the liquid biopsies, not enough DNA in the blood. One of the problems is with, for instance, if you find a very small mass, typically we wait and see. We can't biopsy all of those because most of them are benign. So it would be great for physicians to know if there's an increased risk. If they did our test, our screening test ahead of time, they would know if those masses were increased chance of being cancerous. How would we use our funds? Well, operational expenses, expand our executive team. Actually, last year was our first talk, and Scott Pantel, who's a CEO here, he's now on one of the advisors. So we're getting some great people. We're about to launch. We have a lung trial scheduled for this fall, and we have breast and uterine, et cetera, coming up soon. So we believe it's going to be at Stanford in a couple months. So our advantages, by far the biggest advantage we have is we find it early. An example is colon cancer, stage 1, 95% 5-year survivable rates. Stage 4, a few years ago, was only 5%. So the earlier you find a cancer, the greater the chances. I mean, our test is completely safe. It's a point of care, which is really nice for the physicians. The science, we've got over 20 studies, so it's affordable. The most important thing is we find it early. So we invite you to join us because we think this is the way we can reduce cancer mortality. Thank you, and hope to see some of you in a few minutes.